Good afternoon, everyone. I am Joseph Daniel Irlandes. Together with me is my partner, Stevenson B. Recantin. We will be presenting to you our comprehensive review article entitled Effects of Gamma Irradiation on the Microbial Load of Fruits and Vegetables. The first and foremost keyword that must be taken into consideration in this review is food irradiation. It is a non-thermal food processing method, which means food is processed at near room temperature. So there is no damage to food because heat-sensitive nutritious materials are intact in the food, contrary to thermal processing of food. This is an important consideration, especially in dealing with fruits and vegetables. Moreover, food irradiation can utilize either ionizing or non-ionizing radiation. In this review, the proponents focus on gamma radiation. It is an ionization radiation method that utilizes high frequency, high energy, and short wavelengths to eliminate microorganisms. It is usually applied for shelf life extension, insect disinfestation, delay of ripening, and reduction of microbial load. So why do we need to irradiate fruits and vegetables? They are often cultivated in open areas with potential exposure to eccentric pathogens via soil, irrigation water, manure, wildlife, or other sources. They pose a risk of contamination. Additionally, these products are frequently ready to eat. Consumers only minimally process these foods before eating, which does not guarantee their safety. More details about microbial load and their sources would be discussed as the presentation progresses. Nonetheless, the proponents decided to proceed with this comprehensive review in order to assess the effectiveness of gamma irradiation as a cold sterilization treatment for fruits and vegetables in reducing their microbial load. It also seeks to add to the growing knowledge of the advantages of gamma irradiation that may help producers understand its effectiveness in making sure that their produce is safe for consumption and can remain shelf stable for a longer period of time. The largest pathogen family connected with food is the Enterobacteriaceae, which is frequently included in microbiological criteria and whose presence has historically been associated with food cleanliness and safety. As mentioned beforehand, organic fertilizers, irrigation water quality, and soil contamination all contribute significantly to contamination. From the literatures the proponents were able to access, there were four main classifications of microbial load found in fruits and vegetables. The first one in the list are bacteria. Some of the widely known bacteria that cause severe effects to human health are Salmonella and E. coli. Unsatisfactory levels of microbiological quality are those levels which are beyond the acceptable microbiological limits, which are resulted due to poor hygienic conditions or handling practices. Consequently, further processing and treatment must be done in order to remove harmful bacteria residing in the fresh produce. Nonetheless, bacteria are responsible for infections and spoilage of food. Moving on, post-harvest fungal infection of fruits and vegetables is mainly caused by fungal pathogens that can be harmful to both human and animals as they produce mycotoxins. Post-harvest diseases in fruits and vegetables are serious problems that results in loss of a large percentage of crops. They mostly spoil plants and fruits with low pH such as lemons. The following classifications, namely virus and parasites, are closely linked with the reasons to why they dwell on fresh produce. They only have different families as well as their transmissions and hazards. However, it is important to take note that not all parasites are considered as microbial load. There are only specific types such as intestinal helminths and protozoa. Table 1 in front of you shows the common etiological agents found in fresh produce. Etiological agents are those responsible for contributing to diseases or severe conditions. Uh, this will be not our main focus because the main focus of our review are the effects of the gamma irradiation to this microbial load.
In the irradiation of fruits and vegetables, cobalt-60 is often used instead of cesium-137 as the source of gamma irradiation because cobalt-60 is insoluble in water. This makes cobalt-60's environmental and health risk lower compared to soluble cesium-137. Examples of fruits or vegetables that are irradiated are maize grains, cherry tomatoes, red chilies, and eggplant. Different types of microorganisms may be found in each fruit or vegetable, such as Aspergillus flavus and Listeria monocytogenes. Heat and additives, such as essential oils and acids, produce synergistic effects. In other words, when combined, they produce better results. Gamma irradiation paired with additives also help increase radiosensitivity, where lesser dosages kill the microorganisms. Example is the aflatoxin producing microorganism Aspergillus flavus, which is found in maize grains and is responsible in the rotting of the maize grains. When irradiation alone was used, 4 to 5 kilogray was required to kill the A flavus in the maize grains. But when the maize grains were heated prior to irradiation, the dosage required decreased to only 0.5 to 1 kilograys. The, pre- the preheating of the maize grain weakened and deformed A flavus's structure. Next slide, please. Gamma irradiation, carvacrol, which is an oregano essential oil, and heat treatment affected B series' morphology. Each picture here depicts the condition of the cells after specific treatments. Fine rod-shaped cells of B series are shown in picture A, which is also the control. Picture B shows the cells irradiated at 1.2 kilograys, and minor effects can be seen in the structure. Picture C shows the cells that were irradiated at a higher dosage of 3.6 kilograys, and some cells were deformed. Picture D shows the cells after being heat treated for 10 minutes at 45 degrees Celsius. The cells in picture E were both preheated for 10 minutes and 45 degrees Celsius and then irradiated at 1.2 kilograys. Next, the cells in picture F were treated with 312 ppm of carvacrol alone. The most detrimental effect can be seen in picture G after 312 ppm of carvacrol and 1.2 kilograys of irradiation were used. In conclusion, the carvacrol disrupted the cell membrane of B series, and the heat treatment caused cell membrane deformation, and irradiation caused cellular injuries. Next slide, please. Cellular DNA is the main target of ionizing radiation. Ionizing radiation can, number one, damage DNA protein crosslinks and alter bases. Number two, they can produce holes, electrons, and excited states in the DNA water solvation, and bulk water surrounding the DNA molecule. Indirect effect occurs when the bulk water is irradiated, which then produces radicals such as OH minus, H plus, and electrons. These radicals diffuse to nearby DNA structure and damages them. On the other hand, direct effect occurs when the DNA itself and DNA water solvation is ionized. Direct ionization darkens DNA bases or backbones. Clustered damage occurs when a base is damaged along with double strand breaks. This type of damage has a greater biological effect compared to isolated damage. When clustered damage occurs, cells are unable to repair themselves and will eventually die. An increase in irradiation dosage also showed an inverse relationship with gene transformation efficiency of plasmid. As the irradiation dose increased, the gene transformation efficiency decreased. The decrease in efficiency was caused by single strand and double strand breaks in the plasmid. Interestingly though, Shiga toxin producing Escherichia coli O157H7 activated mechanisms to survive stress conditions induced by low dose gamma irradiation. The E. coli reduced its histidine biosynthesis since histidine enhances oxidative DNA damage by generating intracellular OH radicals. The E. coli also increased its expression of genes involved in sulfur metabolism since sulfur acts as a bacterial defense against reactive oxygen species. Next slide, please.
Okay, going to the irradiation in the Philippines. The Philippine Nuclear Institute, or the PNRI, is the central institution that offers food irradiation treatment services. Approximately 45 tons of spiced and dehydrated vegetables were treated by them. The institute widely utilizes gamma radiation and electron beams as a radiation source in the treatment process in their facilities. It is considered that food irradiation in the country is still not fully developed and categorized as semi-commercial. However, researchers are making an effort to make food irradiation possible in order to export to the US. The list of food irradiation services of PNRI is shown in Table 3. So these are some examples of the food or agricultural product irradiation services of PNRI. Spices, fresh fruits, grains, bulbs, plant, and the purposes are usually for disinfestation, reduction of microbial load, and sprout inhibition and degradation.
In conclusion of the study, the microbial load found in fruits and vegetables include bacteria, viruses, fungi, and some classification of parasites. The microbial load was mitigated by gamma irradiation, and better effects were observed when combined with additives and heat. The irradiation treatment also aided in shelf life extension, insect disinfestation, and delayed ripening of some fruits and vegetables. With proper irradiation dosage, the gamma irradiation specifically targets the microbial DNA, though it is important to note that significantly higher dosages may start to affect nutritional quality. Therefore, establishing the optimum dosage required is crucial for both safety and quality preservation. With all the factors outlined in the study, we believe that gamma irradiation in the Philippines is possible on the commercial scale once more research and testing is done. Next slide, please. For our recommendations, more research is required on the effects of gamma radiation on sensory and nutritional quality of fruits and vegetables, such as texture and vitamin contents. These two qualities are the most important factors when it comes to consumer acceptability. The article focused on the effects of gamma radiation on the microorganism itself. Consequences of gamma radiation overdose on the sensory and nutritional qualities of fruits and vegetables should be elaborated further to establish a safe ground on its use. Societal acceptance of irradiated fruits and vegetables should also be studied to help tackle hesitancy and skepticism of irradiated food. Next slide, please. And thank you. That is all for our thesis presentation.